Yes, I can maybe start with saying how we process them and give Brian a chance to dig up the QC sheet. So these are all like real life samples that we have processed in Oslo for DNA isolation. So I was very familiar with like the tissue of some of them and how to process them. So like the first two are mosquitoes or like midges. And these are both simply small, so that's what makes them, we use one single individual, very tiny, so that what makes them challenging samples. We, more by chance than by design, power mashed one before we cross-linked it, and the other one we cross-linked first, and then power mashed as the low input protocol says. There was a difference between these two in yield, but they're also not the same size, so we cannot say much here. But both ways of processing the tissues felt okay. Then on number 3, Pitrurus. This is this alga single cell forming colonies in their own slime, which I was hoping would resuspend a bit in the PBS, but that didn't work so well. So we still managed to get this through with the rest of the samples. But we didn't do any mechanical disruption since this is not really a tissue. If we need to repeat this in the future, I think resuspending it in something like a sorbitol buffer would be more helpful because that helps break down this sugary matrix. Then we had 2 lichens, Petigeras, which we just did regular liquid nitrogen grinding. Which, just skipping ahead now for a moment, we did the same for Folusta, foliacea, which is bryozoan. Then we had this tiny worm, Meara, who decided that a bit of buffer or a PBS was enough to be mashed, self-mashed, so no tissue processing there needed. And then we had a sponge. Several of you were interested in how we process this. So this is how we managed to get a decent DNA out of it, because every time we tried to grind this tissue mechanically, we noticed that the DNA was very sheared. It was a good yield, but it was just very fragmented. So then our friends at Sanger sent over the squeeze enrichment protocol, which is on protocol IO. I can send it to you if you want, which is basically just soaking the sponge tissue before thawing, like so frozen. You soak it in a EDTA-rich buffer, chop it up a little, and squeeze it to release the cells. And then you collect that liquid, centrifuge to pellet the cells, and then we cross-link the cell pellet. And it seems to have worked quite well. The first 2 were preserved in ethanol, so we just like removed it from the ethanol, just patted it dry with a tissue, and then processed it normally. No wash, no. If it was a bigger tissue, maybe. With this very small insects, I just didn't want to, yeah.
So you didn't freeze by the liquid nitrogen for insects?
No liquid nitrogen for the insects, no. Like it would be just power masher.
Without any liquid.
So in the one that we first cross-linked, so that did dry in the tissue and then directly into PBS and then added the fixation or the cross-linking buffer. And then we power mashed it inside this buffer. This is what the low input protocol says. The one that we tried power mashing before, we just put it dry in the power masher tube and then held the tube on the dry ice and power mashed it, left the pestle of the power masher inside the tube, so that then when we added the buffer, we could wash it.
Yeah. What's the main problem of using it? To the cell itself?
I mean, sometimes it's just the best you can do when your sample is collected. We were having a conversation this morning in the lab, like, would it be possible to cross-link in the field so you don't have to fix it in ethanol, and you have to worry about bringing liquid nitrogen to the field and this kind of things. Sometimes you can just get things in ethanol when they are collected, if there's no other option. And then mostly it works. Sometimes it makes the tissue a bit more difficult to handle, just in terms of the mechanics. It gets a bit more difficult to cryo grind because it can smear, and you don't want that.
Yeah, I heard the comment it gets more rubbery.
Mm-hmm.
Yeah.
It's very tissue dependent.
Yeah.
With this, I mean, these don't need brute force, so this is not an issue here so far, and just from the handling point of view.
But if you're doing it, and if you can, if you have a kind of a normal tissue, an organ or something like from a fish, try to chop it into smaller pieces.
Yeah.
So you have a large volume of ethanol piercing, right? You can't put a chunk like that into a small volume of ethanol.
And another thing that tip from Scooter, actually, from the email lab, when you put insect or something that has outer shell in ethanol, it's a good idea to squeeze it lightly so that you break the carpax or whatever the chitin. So the ethanol would penetrate the inner organs and saturate the tissue much faster. So I've seen some results in the DNA extractions and the quality of the DNA. When it was squeezed, it was much higher in the family weight DNA as compared with just the ooze. You put it, so what they do, they collect Malay traps in 96 volt weight, and then they use 2550 and then we put about inside them.
So the first mosquito was our natural before, was being given without any previous,
Just try. I mean, it's the same principle as cryogramming, like any other, like normal input tissue, right? So I think that's where the idea was to try it out. Felt like it was more ground than the one that we did crosslink first. I was a bit worried of how much was on the vessel, but we were able to wash it so to recover almost everything, I think.
And so you took the tip close to the vessel and you just walk around the buffer around that.
And this is the good thing with ethanol, right? As long as you have preserved an ethanol sample, you also have a bit more time when you are handling it for the prep, right? Because it's kind of well preserved. You don't have to be concerned about this keeping it cold and all that. Okay, work on ice or something, but if you can, but it's not suitable for a prep process.
Yeah.
Yes. On the inside of
And ethanol is actually very good preservative.
Ethanol was tested for vertebrate gene project. We tested different preservative species, and we found that ethanol was absolutely second best after liquid. So I know that some technologies do like to take tissues and ethanol for safety, but so far, so good.
Yeah, but it's our way to start. Of course, there are a few that we didn't QC Q4, just because so from the first QC, started at little material, which is not unexpected, the two mosquitoes and the worm. So we skipped PC2 for this one, but for the other ones, you can see it. So the initial PC and PA are the lichens. FF is the bryozoan. HF is the alga. GB is the sponge, and I think that's it. The other one we didn't do. There's 1 and 2. It's because we split the reaction in 2 because the pellets were quite big. So will look quite good.
The ones which failed, which are
These are not failed, these are just we didn't do QC2 because they're so small. So we had maybe 2 to 500 nanograms.
Yeah, that's it.
Ah.
This is the tender.
For sure. These are very, very different. I mean, we didn't measure the tissue, so it would also not be a fair comparison. I like that from very tiny to, and it's also, for example, the sponge, we used like a tiny piece of tissue and we had like probably the highest yield from all of them. It's like it's can vary a lot. For example, for the alga has a large volume but little cell in this slime, so you'll have a low yield. The lichens have no idea what's in there, so we just took an amount of tissue that we thought was manageable and worked with a pellet of a size that was manageable.
And you use the low input protocol for this samples?
We used the low input volumes for all of them, and then for again based on pellet size, we decided how to aliquot and which one needed to input estimation and so on. But we based that mostly on looks on pellet size and how it was to handle. I mean, some we carried over debris quite a few steps. One is still brown. So in the initial steps, those were the ones with the biggest debris, and actually something that we discussed because they had still a few chunkier pieces that would sediment quite fast, and then it was difficult to even aliquot it in 5 and 10% are very estimated. So one thing we said is maybe next time we just are happy with what stays more or less in suspension and can be aliquoted, and what sediment bottom so quickly it's probably best to leave behind. But yeah, no, more or less we could handle all of them. Maybe it was the alga that because that one when pellet and it soaks up the buffer and just takes up a lot of space. So it was difficult to remove the buffer. Maybe it was that one. Also to place, it was difficult to place. Yeah, because it was still a very connected. Yes.
Could you tell me more about sponges? You mean you let it go?
So take it out from the dry ice or wherever you're keeping it, and then put it on a weighing boat, and immediately added this L1 buffer. Well, we didn't have L1 buffer, so we just used the closest possible, which was the EDTA, which protects the DNA while the sample thaws and you handle it. So it's a sponge. You pipette a few milliliter of this buffer onto the sponge, soaks it up, thaws, and then you can handle it without worry that the DNA will damage further. So we know that this works for DNA integrity. We don't know if it works for nuclei integrity. We are hoping, but the idea is to release the cell from this, the taxonomist, forgive me, skeleton or whatever it is that a sponge has. So then once you have squeezed it, you pipette the liquid back out. So we try to use forceps and spatula, just whatever makes sense. Pipette the liquid out again, and then spin it down. Yeah.
So at the beginning, there were a lot of tricks done here, and all samples behaving slightly different. But with some experience, you kind of make an educated guess what to do with it. Leave something behind is maybe better than trying to continue with everything. So this comes with experience.
And I have to say it's a big help if you have. So I have worked with many of these for DNA isolation, so I was not completely shocked by some of the tissues. That helps if you have the chance of doing both, or to talk to the person that did the other one. That can be very helpful.
Yeah, that's a great thing.
Yeah, that's the example that Martin showed earlier with the several libraries. So that was a plant. And there we do nuclei isolation first, and then cross-link the nuclei. And then they can be treated same as you have done. Oh, well, not you, but the others that have used the Arima.
Yeah.
Yeah, and then, I mean, we have been doing both. And I think now we're quite happy to go forward with isolating the nuclei first and cross-linking the nuclei.
Yeah.
It's a bit less messy as a protocol, less time-consuming. There's different nuclei isolation protocols. There's kits. Personally, I really like the Royal Botanic Garden Edinburgh protocol that was developed for Sanger or for DTOL for challenging plants. Since we often don't know which plant is going to be challenging beforehand, it's just an extra wash step with the sorbitol. So we just I like to use that one, but both also the one recommended by Papayo works for us very well. I can send it around